welcome. I'm going to allow for things to calm down just a second, because you might have noticed there's a few other events happening right around us, this is the first time that's happened, so you all get a special kind of chaos event outside. But thank you for being here. My name is Dion Rossiter. I'm an Executive Director of Science at Cal. Raise your hand if you've been to a Science at Cal event in the past. Oh, returners. And if this is your first time at a Science at Cal event, raise your hand. Yes, nice to have you. We hope that you come back and visit us again. I'm going to borrow this and then hand it to you. I'm going to start today with a land acknowledgement. That's typically how we start all of our programs, so if you just give me a moment. We recognize that Berkeley sits on the Huchun territory, the ancestral and unceded land of the Ohlone people, the successors of the historic and sovereign Verona Band of Alameda County. This land was and continues to be of great importance to the Ohlone tribe and other familial descendants of the Verona Band. Every member of the Berkeley community has and continues to benefit from the use and occupation of this land since the institution's founding in 1868. By offering this land acknowledgement, the Berkeley community not only recognizes the history of the land in which we stand, but also recognizes that the Ohlone people are alive and flourishing members of the Berkeley and broader Bay Area communities today. So again, thank you for allowing me to make that acknowledgement. In 2008, Science at Cal was envisioned as a unifying effort to raise public awareness, understanding, and appreciation of scientific research at UC Berkeley. To realize this vision, we engaged the vast Berkeley science, technology, engineering, and mathematics, also known as STEM, community to foster creative collaboration amongst campus and community-based groups who share our commitment to equity and inclusion in STEM education and STEM careers. Science at Cal connects researchers with diverse community groups of all ages and backgrounds for science engagement and science learning, accessibility, inclusiveness, creativity, and innovations, all hallmarks of Science at Cal events, which reach tens of thousands of people annually. Throughout the year, Science at Cal presents ongoing free outreach programs on and off campus. We help promote all related efforts within campus, and we create new programs and initiatives at Berkeley and in the community. This broad scope of activities is made possible by Science at Cal's dynamic network of campus alliances and valuable community partners. So, as you can see here, back here, we do a ton of things. I'm going to give you some highlights of what's coming up in the next month so you can join us for other events. I want to thank Berkeley Community Media. These are the folks who are filming this event, and we're going to give them a round of applause for being here. If you have any video needs, please look them up, give them a call, talk to them after the event. They're here to help. So thank you to Berkeley Community Media. I want to thank the Science at Cal Advisory Council. I want to, I want to thank the UC Berkeley Executive Vice Chancellor and Provost for supporting Science at Cal. I want to thank the Lawrence Hall of Science, who manages Science at Cal. We are employees of the Lawrence Hall of Science, so it's a beautiful day. Head up there if you have some time. And I want to thank donors. That includes anyone who's given in the past or anyone who gave today for this lecture or who will give in the future. Of course, we want to make sure that all our events are accessible to the folks who need them. So we make our programs free. That's very important to us, but also we ask that people, if they can, donate so that we can continue to do that for folks who need it. So that's a QR code, and there's one out on our welcome table as well if you'd like to donate. We have two virtual programs that are coming up. One is on animals, that is also on animals, so October was our animal month. All about animals. This is in 2 Thursdays, and this is at noon, and this is in collaboration with Berkeley Lab. So if you want to just watch from the comfort of your home while you're eating lunch, please do that. We also have the Road to Vehicle Electrification in November on the 17th, also virtual. It's like you don't want to push it twice because then it'll skip. Here we go. We also have a program that's in Spanish. These are held every single month, and our one for October is called Genome Spilled the Beans About Bats. And that, again, is going to be in Spanish. So if you know of any Spanish-speaking folks, if you speak Spanish yourself, or if you want to share with the community members, please do. And this one's a little closer to Halloween, so it's exciting that we get to talk about something spooky, like bats, with the Oakland community. This event takes place at the Fruitvale Oakland Library. We will also be at Dia de los Muertos Festival in Fruitvale, so come on out, join us, meet some scientists there. This will be on October 30th, so we'll have some scientists, and we'll be out with the Lawrence Hall of Science. And I think that might be it. So I'm gonna do an intro, because you're not here to hear me speak. Before I give my intro, I just wanna remind you that if you have to exit anytime during the event, try to do it quietly, where you come in and come out as quietly as you can, because the room's acoustics, you might have already noticed, are a little funky. I've asked Becca Tarvin if she'd like questions during her talk, and she said that's totally fine. So you're more than welcome to raise your hand during the talk, and she's gonna repeat the question and then answer it. And then we'll have an extended Q&A at 12, where then we'll do lots of Q&A time. So if you want to save your Q&A till the end, that's fine as well too. So that's all the logistics things. Rebecca, or also Becca D. Tarvin, obtained her BA in biology from Boston University in 2010. She worked abroad conducting field work in Ecuador, and then obtained a PhD in biological sciences from the Department of Integrative Biology at the University of Texas at Austin in 2017. After a year-long postdoctoral research position at UT Austin, she conducted another abbreviated postdoctoral scholarship position as a Miller Fellow from 2018 to 2019. Becca's PhD and postdoctoral work focused on the ecology, evolution, and molecular biology of poison frogs from Ecuador and Colombia. Currently, Becca is assistant professor in the Department of Integrative Biology and an assistant curator of herpetology at the Museum of Vertebrate Zoology here at UC Berkeley. Her lab studies the evolutionary mechanisms underlying adaptation and diversification, with a focus on animals that acquire chemical defenses from their environment, aka poison frogs, which we're going to hear all about. So I am going to hand things over to Becca. We will switch up our presentations. She'll turn on her mic. You want to do that?
Yes.
I'll hand her the pointer.
Thank you. Okay. Can you all hear me okay? Yes, okay. And I just want to make a quick announcement. I know there's admissions activities going on. If that's what you're here for, you're in the wrong place. And those are happening, I believe, in 2040 and 2060. But if you want to stay and learn a bit about poison frogs, I certainly welcome that. All right, so today I'm here to talk to you about nature's toxic defenses. And up here on the screen, you can see an image, a photograph that was taken in the 1970s of a member of the Embera indigenous tribe who live in northwestern Colombia. And you can see them in the act of poisoning a dart. And this is, in fact, how poison dart frogs got their name, from this very practice that's been done for a very long time by the Embera indigenous group. And this frog here, this is called Phyllobates terribilis, or the golden poison frog, and it is very toxic. Just simply wiping a dart on its back is enough poison to kill a large mammal. Okay, in the 1970s, when Western scientists from the US went to South America to study all of these interesting chemicals found in colorful frogs, they actually used a method where they tasted the frogs. So this is just words from one of the publications from 1976, where they write, initial bioassays included tasting the back skins of the frogs in the field, though they also used subcutaneous injection of extracts into laboratory mice. So this was work primarily conducted by a man called John Daly, who at the time was employed by the National Institutes of Health. And he spent decades going to South America and trying to figure out what are these cool chemicals that all these colorful frogs have. And indeed, you know, this is the footnote here under tasting. They say, in our experience, a frog with a disagreeable taste usually contains a pharmacologically active substance in the skin. So this is, in fact, you know, often you eat bitter compounds, and those tend to be kind of active substances, right? They taste bitter, they're interacting with some sort of protein on your tongue. They say the field test seems safe enough in the case of Dendrobates, which is a genus of poison frogs, but we do not recommend tasting those of species of Phyllobates, which was, in fact, the picture of the frog that I showed on the previous slide. And that's because they might contain dangerous amounts of toxins. So this is a particularly potent neurotoxin that is only found in Phyllobates poison frogs. They note here, unfortunately, there's no a priori way of knowing which species is a Phyllobates. So we now know in the 1970s, it wasn't as well known, but now we know at least which species these are. Yes, is there a question? That's a good question. I never really thought twice about that. But yeah, this is Charles Myers. So the two people that went down from the US to investigate these compounds were John Daly from the National Institutes of Health, and this is Charles Myers, who was a herpetologist at the American Museum of Natural History. No, this is just if you search Charles Myers, this is the picture that is for some reason on the internet. Yes?
So, when Myers and Downey tasted the frog's back skins, do they get poisoned by them?
Okay, the question is, when the researchers tasted the skins of the frogs, did they get poisoned? So that's what they're warning against here. So these frogs are actually quite small. If you look at this picture, this is actually a relatively large poison frog. And so you can see maybe it's the size of, half your finger, but most of the poison frogs are half of that size. And so the amount of toxin that a single frog carries is usually not enough to harm a human, except in the case of this very specific toxin called toxin. And that's because it's a particularly potent toxin. Yeah, and that's why this one is dangerous and many of the other ones are, it may not taste great, but you're not going to die. I still would not recommend licking any frogs. Other questions? Okay, so why in the world would a frog be so toxic? Well, we know, first of all, that poison frogs are unique in that they don't produce these toxins. They actually obtain them from their food. So whenever you see a frog in a zoo, a poison frog, they're not actually toxic. And that's because they're away from their normal food source, and their food source is where they get the toxins. In the case of toxins, it's suspected to be a beetle that these frogs consume. And how are these toxins, like toxin, so potent? Well, toxin is a type of neurotoxin. And that means that it affects the nervous system. The nervous system is composed, for the most part, of cells called neurons. And neurons communicate using electrical signals. So they rely on ion channel proteins to send and receive messages. So these are special proteins that govern the flux of ions across cell membranes. So ions are charged particles, like sodium, for example. So if you're familiar with an action potential, these are electrical signals that flow along cell membranes that are governed by the movement of sodium ions and potassium ions. And the way that these ions move in and out of cells is through these ion channels. So these are very critical proteins that you and I rely on every second, every millisecond, as do many other animals. Even bacteria have these sorts of channels. And I'm going to pass around, I have a 3D printed voltage-gated sodium channel. So this is a specific type of ion channel that governs the flux of sodium ions. And when you see it, you'll notice that there's a blue a blue molecule stuck in there, that's toxin. So you can see where it's bound. Okay, so ion channels are these critical proteins that you and I have, that frogs have, that snakes have. Every animal has these. And they allow for the flux of ions. So when an action potential occurs, this is because voltage-gated sodium channels sense the accumulation of sodium ions outside the cell membrane, and then they open their gate and they allow sodium to enter into a cell. And this results in the propagation of an electrical signal, which causes, for example, in the case of a motor neuron, the contraction of a muscle. And so when there are toxins present, neurotoxins like toxin, they're messing with the function of these proteins. In the case of toxin, we know that they are super activators. So they actually keep those pores open, and they allow for continual entrance of sodium into the cell. So while normally sodium would enter once, a signal would go down, they keep allowing sodium into the cell until there's no more sodium outside of the cell, and then there cannot be any further signals. So this is a type of paralysis called spastic paralysis, where you might see sort of contraction of muscles and then there's no more movement. And this can actually be quite lethal. So this is one example of a type of toxin. There are, of course, many toxins with many sorts of action on our body. But in general, we can kind of categorize them broadly into two categories. Neurotoxins are these types of compounds that damage or affect the nervous system, which is in control of your body. But you can also have toxins that are directed towards specific aspects of the body itself. Okay, so these are called, again, neurotoxins, affect the nervous system, and then anything else is usually named for what it's messing up. So hemotoxins are named for messing with blood, and cytotoxins, for example, are targeting cells. Okay, so as I mentioned, we know that neurotoxins affect the nervous system. And we know also that the nervous system is generally composed of the central nervous system, which is the brain and the spinal cord, and the peripheral nervous system, which are the sensory and motor nerves. And most of this tissue is composed of cells called neurons. Of course, there are many types of neurons, but we can kind of think about toxins acting on information flow in the nervous system. So we know, for example, that in the peripheral nervous system, that's where we acquire information from the environment. The central nervous system is where that information is processed and interpreted. And the peripheral nervous system, again, is where signals are sent from the central nervous system out into things like muscle contraction and movement. And neurotoxins can affect all three aspects of the nervous system function. So for example, you could experience loss of feeling or pain. And this is where the signal from the sensory system to the brain, and sometimes actually from the brain to the muscles, is being interrupted. And a great example of this, toxin. So I have a colleague who is from Colombia who I asked, have you actually experienced this toxin? And so when you work with Phyllobates poison frogs, you have to take precautions. So you wear gloves, you're very careful about how you're handling them, but mistakes can be made. And he said one time he had all these precautions in place, and all of a sudden his face started going numb. So somehow the toxins had gotten onto his face. He's fine, it was temporary, but that's, for example, the sort of experience you might have in a toxin that affects the sensory system. You could also, yes? Sorry, a question here up front and then in the back. Yes, go ahead. Okay, the question is, because neurotoxins can affect the sensory system, could they numb the whole body? And it depends on the toxin. So you can be very precise about how you apply a neurotoxin, and if it's not easily transported around the body, then it could be either local anesthetic, for example, like who's had lidocaine from the dentist, right? That's like a toxin that's blocking pain receptors in the mouth, so that's a local numbing agent. And you can also have, you know, a sort of toxin that would numb your whole body. That's quite dangerous, right? But many of these toxins are actually similar to the way that drugs work. Mm-hmm. And in the back, was there another question?
Species of frogs that even if you have these precautious gloves, that it actually still seeps through after like a period of time, right? Is it like a certain, like, I don't know, if it's latex or whatever it may be that is absorbed through, or is it just general, it can seep through the gloves or precautions that you take?
Yes, okay, so the question was, are there cases for frogs, for example, where there are specific toxins that can even go through gloves? And so it depends. So all of this depends on the chemical structure of the compound and what sorts of solubility it has. So some compounds are very good at passing through barriers and others are not. And so in the case of batrachotoxin, for example, batrachotoxin can easily go through cell membrane. So the cell is, you know, it has a kind of lipophilic layer, so it's able to go through things that you might think are impermeable. But that being said, most nitrile gloves that we use in the lab, I don't think batrachotoxin would go through that. But there are, for example, if you're dissolving a toxin in a specific buffer, sometimes those buffers, you might be using it in order to make it more soluble in something else. Sometimes that can create a situation where that buffer would make it more soluble through your gloves. So yeah, I don't know of a specific frog species that you might be thinking of, but these are all things you have to be careful of when you're working with toxins. Yes? Okay, the question is, if you poison a dart with this toxin, how long is it poisonous on the dart? I don't know for sure. Some of these compounds are pretty stable. For example, the compound that I do know some details about is tetrodotoxin, which is another type of neurotoxin that actually targets the same protein. Tetrodotoxin can be pretty stable at room temperature for, it must be at least several weeks. If you put it in the freezer, it can last, you know, 10 years. I don't know specifically about batrachotoxin. Yeah, it probably depends on, you know, are you storing it in a pot of bacteria or did you put it in the fridge? Yeah, I imagine they must use it fairly quickly. Any other questions? Yes?
Hunters that are using these, how do they carry them around?
That's a great question. So the question was, how do the hunters kind of protect themselves from the toxin? And I don't know. I would have to go into the anthropological literature. I'm sure it's known. Great question. Yes?
Do hunters use them as weapons to hunt down animals?
The question was, do the hunters use these darts from poison dart frogs to hunt animals? Yes. Yeah, they have they use a blowgun. So what you saw in the first part was a dart, and so they basically take the frog. In the case of Phyllobates terribilis, they're so toxic, all you have to do is wipe the dart. But in other cases, they might actually stress the frog. So often frogs won't release their toxins unless they feel like their life is in danger. And so they might hold the frog and make it feel uncomfortable and then wipe the toxin, or they might mix plant toxins and frog toxins and use that. But yes, it's for hunting. Yes?
If you eat an animal that was killed by a poison dart, then could the poison get into the...
This is a really good question. So, okay, so if you're using a lethal poison to kill something, is it safe to eat that animal? Which is a really good question. They must eat the animal. Probably if you're cooking the meat, it would break apart the toxin. But yeah, that's a good question. You know, people would have to be careful, and I would have to ask them, you know, exactly what they do. Great question. Any other questions? Okay. Okay, so we were talking about how toxins can mess with your nervous system. And I was mentioning that they can affect kind of this information flow into the brain. They can also affect how your brain functions. So a toxin that affects sensory and signal processing could result in brain damage. It could cause memory loss. It could also have epileptic effects, so seizures. An example of this is a toxin called domoic acid. And this is actually produced by an algae, but in marine environments, it can be bioaccumulated by filter feeders, such as clams, but also the Dungeness crab, which maybe you've eaten before. And actually, in the 1960s, there was an algal bloom where many of the crabs had bioaccumulated domoic acid, and the seagulls had eaten the crabs and were acting erratically, and you know, it seemed like they call it a seabird invasion. So the people of Santa Cruz were alarmed by the strange behavior of all of the seagulls, and this is presumably because they had been affected neurologically by domoic acid. And in fact, in fact, this is purportedly what led Alfred Hitchcock to tell his story of the birds. And so if you're not familiar with this movie, this is a really great movie to watch. It was filmed in Bodega Bay, so you can watch the movie, and if you haven't been to Bodega Bay, go check out. You'll probably recognize some of the filming sites. Still a great movie. Yes?
Do domoic acid make the birds go crazy?
The question was, does domoic acid make the birds go crazy? Yes. Yeah. Any other questions? Okay. So we can also see toxins that affect muscle control. So again, this is information from the brain to the peripheral nervous system. And an example here is botulinum toxin, or as you might know it, Botox. And so this is a toxin produced by a bacterium that we use to temporarily erase wrinkles because it paralyzes your muscles. Kind of scary. Okay, so those are neurotoxins, and I'd just like to introduce you to two different types of toxins that affect the body as well. If you're uncomfortable with blood or snakes, you might want to zone out for the next 30 seconds because I want to show you what happens when you mix viper venom with blood, okay? So a lot of snake venom affects blood clotting processes, and sometimes it can make blood clot very quickly. Other times, it can prevent blood from clotting, and both of those are not good. So this is a pretty effective sort of toxin. And can I get a show of hands? How many of you have been stung by fire ants? Okay, so you know, it's not a great experience, but if you're interested in toxins, it's pretty fascinating. And I apologize, it's a little gross, but I like it as an example. So when a fire ant stings you, it injects a venom that's cytotoxic. And so cytotoxic refers to the fact that it destroys the cells, so it kills the cells. But when you get stung, and if you've had this experience, it almost looks like a pimple that can be popped, that's maybe full of pus, but it's not. It's not that. It's just a cluster of dead cells. So if, you know, this is something, if you ever do get stung by a fire ant, you won't forget. Okay, so just to summarize, we've just discussed kind of two broad categories of toxins: the neurotoxins, which affect the nervous system, and then other toxins, which are usually named for their specific mode of action. So why might an animal be toxic? Well, the most common reason that you're probably familiar with is an anti-predator defense. So lots of animals have this to ward off predators. Sometimes they have brilliant colors or shapes for predators to remember that bad experience. And a classic set of experiments for biologists is this demonstration where blue jays were fed monarch butterflies. And as you probably know, monarch butterflies eat milkweeds. They sequester cardiotoxins, so a type of neurotoxins that affect ion channels in the heart. And they also are emetic, which means that they produce vomiting in something that eats them. And so this poor blue jay was naive. This is a blue jay raised in a lab environment, had never seen a monarch butterfly in its life before, and it certainly remembered the experience, and it did not return to consume it. So lots of animals have these types of toxins that ward off predators. They can also kill parasites. Yes?
Remember the monarch butterfly getting eaten by them and vomit. Will they avoid monarch butterflies or some other mimics that butterfly that mimics the monarch butterfly?
Yeah, very insightful question. So the question was, if a blue jay has this experience where it vomits after eating a monarch butterfly, will it remember that? And will it also avoid other butterflies that mimic monarchs? And that's exactly how it works. So if in the future, this specific blue jay would avoid eating a monarch butterfly, but also other butterflies that look like monarch butterflies. Yeah. And that's how mimicry can be effective in toxic animals. Okay, so other animals you might be familiar with that are toxic often use toxins or venoms to capture and digest prey. And I mentioned not just capture, but digest, because in the case of snakes and actually many animals that use venom to kill prey, these proteins are often evolved from digestive enzymes. So they are types of enzymes that help break down flesh. And so not only can it harm that animal, but it can also be useful. For example, if you're a spider eating some kind of fly, right? So they inject venom into the body of something, break it all down, and then it's nice and easy to suck back out. So snakes, of course, do this in a different way. They just put the whole thing in their mouth. And the last major reason is something maybe you don't think about so often, which is that some plants and animals actually use toxins as a matter of competition. So for example, if a plant is growing in a specific area, it might distribute a toxin in the soil that prevents other plants from growing in the same place. So that's useful for the plant. And some animals do this as well. So the other image up there is a cluster of toad tadpoles. So if you've ever seen toad tadpoles, first of all, they're super cute. But they also tend to be communal. So toad tadpoles have social behaviors where they hang out together, but they also produce toxins just like the adults do. And those toxins can be harmful to, if there were a tree frog tadpole there, it might be susceptible to that type of toxin. Okay, so how do animals get these toxins? Well, as I mentioned, in the case of poison frogs, for example, they obtain it from their food. This is the case for many types of animals, often things you don't think about. So this is a picture of a nudibranch, it's a type of marine slug that lives in tidal pools. In fact, if you've never been tide pooling in California, we have an amazing diversity of nudibranchs. And this is a species that you can see on the coastline. They're really gorgeous. This one's called an opalescent nudibranch. And they're actually really small, so they're hard to find. This species is about 1 centimeter, so like between half an inch to an inch long. And they do this super cool behavior where these nudibranchs are actually specialists on different types of cnidarians, so relatives of anemones. And as you might know, anemones and jellyfish, which are both different types of cnidarians, they produce stinging cells, right? So those stinging cells are actually tiny organelles that have a harpoon and venom wound up at high pressure in a little tank. And these sea slugs, they are able to eat. So this is a picture of a hydroid. It's a type of cnidarian that nudibranchs consume. And the nudibranch will actually graze on that like a tiny cow, and it will take up those stinging cells into a cnidosac. So these are appendages on the back. I'll get to your question just in a second. These are appendages on the back that are actually, they're called digestive diverticula, and they are part of the digestive system. And at the very tip, there's a little sphincter and then something called a cnidosac. And so when these sea slugs consume stinging cells, they put the stinging cells up into the cnidosac, and they keep them there. And they basically repurpose and they reuse the stinging cells against their own predators. So this is a really cool case of sequestration. Yes?
The anemones to the type of anemones.
From the what?
The standard anemones to the type of anemones.
So, okay, so the question is, what's the difference between getting this from anemones and the specific one here, the hydroid? Yeah, so anemones, I don't know offhand if there are animals that sequester their stinging cells, but cnidarians are a huge group of animals that all possess the same type of stinging cell. And so they may have different kinds of stinging cells, and anemones, because they tend to be large, may have larger stinging cells that might be too big for an animal like a tiny nudibranch to consume. Yes?
Get that through their digestive system up through that thing without firing it.
Thank you for asking that. I feel like I planted that question. So the question was, how do nudibranchs obtain a highly pressurized stinging cell without exploding it? And so the answer is they don't. They don't and they do. And the reason is, so this isn't very well studied. There's been a couple papers about it. But there's some evidence that nudibranchs have specialized mucus. Thank you. Specialized mucus that has these spindles that kind of entangle any sort of, if the nematocyst, the stinging cell, fires, they have some sort of protection against it. So the spindles might like wind up the harpoon. They also have keratin, so the same type of compound in your fingernail, lining their mouth. So if it happens when they consume it, they're also protected. But the bigger question is, how do you get it up into the cnidosac? And so what we think, and this is speculation at this point, but the idea is that actually the ones that go into the cnidosac are immature cells. So cells that are still in the process of being pressurized. And the fascinating part here is, okay, well, how do they become mature? And again, total conjecture. This is something we're interested in studying. But we think what's going on is they take up an immature cell into the cnidosac. And then actually, if you look at dissection histology, you can see that those cells slowly die. The cells that take up they basically engulf the nematocyst, and then that cell kind of shrinks and dies. And during the process of cell death, there's a change in the pH, and they think that this lower pH is what allows the nematocyst to mature, to finish pressurizing itself. So this is all hypothesis, but that's what we think is going on. Yeah. Any other questions? Okay. All right, so this is my favorite way to get toxins. This is also how the poison frogs do it, how the monarch butterflies do it, but many other animals synthesize it. So many snakes do this, ants do this, bees synthesize. So this is when you're producing usually a protein, but it can also be a steroid, for example. But you're using your own biological pathways to produce this toxic compound. And the third major way is actually obtaining a toxin from a symbiotic partner. So who knows what this is? California newt. Yeah. Yeah, so this is a type of salamander that lives in our backyard. Usually you can find them when it's raining. So if you have not seen this salamander, I urge you, when it's raining, go to Briones, go check out the cattle ponds, because you'll see literally thousands of these animals. They hide when there's no rain. They hide because they require the moisture in order to survive. But what's fascinating about these salamanders is that they're highly toxic, but they don't make the toxin, and they don't eat the toxin. They have bacteria living on their skin that produce the toxin for them. So not something that's as well studied, but definitely occurs in a number of situations. Yes?
To add that both Tarica turosa and Tarica granulosa are at the UC Botanic Garden Asian Pond. So that's for people who don't want to try to find reunities, you can just go up to the Botanic Garden.
Thank you. Yeah, so that was a comment pointing out that actually even year-round at the Botanical Garden on campus, you can find both, there's two species that look very similar of this salamander, and you can see them in the Botanical Garden in the Asian Pond. So go check them out if you haven't seen them. They're really cute. Yes?
I don't know, but they've closed South Park Drive and Childers for many years because the news right now, if there are any left, I don't know, the droughts pretty bad, are up in the woods across the road, and in the winter, fall, if it rains, when it rains, I've seen thousands crossing, and I've also seen thousands of them crushed before they close the river.
Yes.
Somehow in that golf course, they find water. And I guess you have to go all the way to Wildcat Creek, but that's pretty easy to get to. But it's best if it's pouring in.
Yes. Yes, okay, so that was also pointing out that you may have experienced the closure of South Park Drive in Tilden, and the reason that's done every year seasonally in the wet season is because these salamanders come out from all their hiding places and they walk across the road, and in the past, when that drive was not closed, many of them, thousands of them died because of cars would run them over. And so you can go, especially when it's raining and especially at night, that's a great place to find these awesome salamanders. And yes, you can handle them. Don't eat them. And if you handle them, all you have to do is wash your hands afterwards. So tetrodotoxin is not a toxin that's good at crossing the skin barrier. So you don't have to be scared of them. Just handle them wisely. Just wash your hands afterwards. Did you have a question too? No. Okay. Yes.
The bacteria live on the newt's skin. So, where did the newts get this bacteria initially? Is it from their environment?
Okay. The question is, the newts have this bacteria on their skin, and where does the bacteria come from? And that's something that we don't know. There are different hypotheses. So, like you suggested, it could come from the environment. It could also be what we would say vertically transferred, so from the parent to the offspring. But we don't know. That's an active area of investigation. Good question. Any other questions? Yes?
There might be neurological effects that could even perhaps reinforce memory. Is there something like that known in these other three categories of obtaining your defense?
That's an interesting question. So the question, if I understand correctly, is do some toxic defenses also have neurological effects that can make it easier to remember? Is that what you're asking?
Yeah.
I don't know if anyone's really looked into that, but it's an intriguing idea. Yeah, so some of these, some receptors for neuroactive compounds are expressed in the brain and in the gut, and so you might hear it's called like the gut-brain axis, and people have conjectured like, well, if you're eating really well, that makes you feel really good because of this connection. And we don't understand it very well. But from a mechanistic point of view, if you have a toxin that binds to things in your brain and things in your gut, you can see easily how, and you've probably experienced this where you throw up after eating something bad and then you can't eat that thing that you used to enjoy. And I don't know exactly how that plays into the way the brain functions, but it's an intriguing question. Yes? Okay, the question is, do we know what the salamander gives back to the bacteria? No. Again, this is an active area of investigation. In fact, it wasn't known that bacteria produce this toxin for the newts until last year. So this is completely new in the scientific literature, although tetrodotoxin itself is a molecule that's been known about by humans for millennia. You might know it from pufferfish, right? People have long eaten pufferfish sushi or fugu, and there's this whole idea that it's kind of this risky adrenaline rush sort of experience, and there's a lot of thought that goes into how you might prepare that safely, and it's because in pufferfish, in certain species of pufferfish, you only find the toxin in the liver and the skin, and therefore you can prepare the mussel safely, as long as during the preparation process you don't cross-contaminate. Yes? Yeah, there is that story that dolphins will prick themselves on a pufferfish. So it's not, tetrodotoxin is not really a psychoactive compound. It's more like a compound that could kill you. I don't think it has psychoactive potential. So I'm kind of curious what exactly is going on there. Yes?
On the same line, there is a frog that has a toxin, it's highly toxic in the Southwest, I think, but it's also hallucinogenic if you prepare it correctly. But that also brings up other plants that are hallucinogenic, and that doesn't, I mean, it might be protective because people don't want to have crazy thoughts, but it seems to attract a lot of attention and people gathering them and eating them for that. So it's curious how that's a protective.
Yes, yeah. So the comment was about how lots of plants and even toads have these psychoactive compounds, and a bigger question of why would a psychoactive compound be protective? And I don't have a good answer to that. I just noticed the time, so I'm going to temporarily stop taking questions during the talk and just finish up my slides, and then we can have a longer conversation.
Also, because it's been so active, feel free to go longer.
Okay, yeah. Thank you. All right, so just to summarize what we've talked about so far, this is a fire salamander, by the way, Salamandra salamandra from Europe. How do toxins work? Well, they can be neurotoxins that target the nervous system, or they can be other types of toxins named for their mode of action. Why might you have toxins? To reduce or prevent predation, to capture and digest prey, or to compete with others. And how do you get toxins? You can sequester them, you can synthesize them, or you can obtain them through symbiosis. Okay, so going back to these ion channels, and hopefully everyone got a chance to look at the 3D printed model. If not, I'll have it here afterwards. So one common mechanism of toxin resistance is called target site insensitivity. This is a fancy sounding word, but basically it means that if you change the shape of the binding site, the toxin can no longer bind. So you can think about this like a lock and key, where the lock is the protein that's being targeted, and the key is the toxin. So you simply change the shape of the lock, and the key doesn't fit. So again, this is called target site insensitivity, and it evolves very simply through single mutations in the protein target. So for example, just to show you in the schematic, if you change the shape of the binding site in a voltage-gated sodium channel, batracha toxin can no longer bind. You end up with normal flux of ions, normal closing of channel, normal sort of muscle contraction. So this is a mechanistically simple way to evolve toxin resistance. And this is, in fact, what some poison frogs have done to prevent self-intoxication. And so on the top here, this is a two-dimensional model of a voltage-gated sodium channel. So I showed you kind of a mechanistic of what it looked like. If you looked at it, you can see it's kind of this cluster. It's got a pore in the middle. But basically, this is a type of protein that sits in the cell membrane. And so this line here, this is supposed to be the cell membrane. And you can see the protein itself is this one long string of amino acids that consistently wraps through the cell membrane in and out, in and out. So each of those cylinders it's called an alpha helix. It's a helix of amino acids going through the cell membrane. And it sits there, and a cluster of them form around and create this pore in the membrane. So this is a lot of kind of columns in the cell membrane, and then they can push open and allow ions to enter. And so if you look at the sequences, so if you look at the amino acid sequences, you can see in poison frogs they have some changes that are unique to toxic poison frogs. And that gives us this idea that maybe they have some functional effect on the protein. And in fact, these Phyllobates frogs, we think, have some of these changes. And this is how we might look at it from a scientific perspective. So this is what's called a phylogenetic tree, which is a tree indicating evolutionary relationships, so similar to a family tree. And all of those names there, those are all Latin names of specific species of poison frogs. And what's incredible and what makes poison frogs such a great system to study as a scientist, although it's misleading, not all poison frogs are poisonous. In fact, they've evolved the ability to sequester toxins from their prey 3 separate times. So that gives us kind of a natural experiment to compare poisonous poison frogs with non-poisonous poison frogs. And I've just put this information here by coloring the branches to show you which branches, which evolutionary lineages, and which species are toxic. Those are the ones in yellow. And the ones in black are not toxic. And so what we can do is we sequence the binding site of a toxin, and we look to see, okay, do the toxic ones have changes that are not present in the non-toxic species? And that is, in fact, what we can see. So these are amino acid sequences. These are all single letter codes for different amino acids that correspond to this specific cylinder that goes through the cell membrane. And you can see that often the changes are present in these toxic species, but absent in the non-toxic species. And yeah, just to highlight, this is the location. And that subunit is close to the pore where batrachotoxin binds. And so if you look at Phyllobates terribilis, that's this frog that's used for poisoning darts. They have various changes in these regions that might be involved in resistance to the toxins. Even cooler, or just as cool, I suppose, we know that poisonous frogs don't have a lot of predators, but some of them are snakes. And these snakes are specialized on eating frogs. And if you sequence the same gene in the snakes, they have the same set of mutations. So again, these are proteins that are critical in how nervous systems function. And they are genes that we have, that frogs have, that snakes have, that even insects have. And you can see this amazing evolutionary convergence across millions of years of divergence. But of course, target site insensitivity is not the only way to resist toxins. There are lots of ways to do this. For example, snakes produce these venomous proteins that are injected into your blood. And rodents, which are really great prey items for snakes, and I was amazed to learn this, but even squirrels, which seem like how could a squirrel, not die from a snake bite? But in fact, most squirrels are really resistant to snake venom. And it's because they produce proteins in the blood that bind to snake venom proteins and prevent the snake venom proteins from working. So yeah, it's really cool. And you can even see tracking where snakes, if they eat mostly squirrels, those squirrels will have a high amount of those binding proteins that prevent the venoms. Another cool example that's totally different is bombardier beetles. So these are really awesome beetles that produce precursors to toxins. So they have 2 separate glands where they store the precursors separately. And then when they feel, they have this little hatch here. When they feel in danger, they open the hatches, and both of those precursors mix in this tight compartment that's structurally sound, protected again by keratin, just like the sea slug mouse. And it produces a combustive reaction that explodes at 100 degrees Celsius, so at burning temperature, and it's highly acidic. So not a pleasant thing. Here's a video of it happening. So this is a upset bombardier beetle. And there's a funny story of Charles Darwin one day collecting beetles. So he had 1 beetle in each hand, and he saw another beetle, and he thought, how am I going to get that 3rd beetle? Because he needed all 3. And so he popped 1 beetle into his mouth, and it was a bombardier beetle. And I also love to show this video because my background is with frogs, and I just want you guys to appreciate them as much as I do. And so this is a case where a toad was given a bombardier beetle to eat. And so, of course, the bombardier beetle responded as you might expect. And it was enough. 80 minutes later, I don't know why it waited so long, but it... So the toad decided it had enough, and that was very effective for the beetle, and both of them are fine. Incredible. So that's like a very interesting, both the mechanism of defense and resistance. I don't know how the toads aren't bothered by that. But so this is part to point out, like, you don't have to be toxic in order to resist toxins, right? It's really a question of how often are you exposed to toxins, and is it useful for you to maintain resistance? And so again, this is a voltage-gated sodium channel showing you where those places are where poison frogs have mutations that might provide resistance through target site insensitivity. Well, we also know that flies and mosquitoes also have mutations in this region, and that helps them resist DDT. So DDT was an insecticide that was used a lot in the 50s and 60s. It's currently banned, although it is used in other countries. And it's very effective because it particularly affects the voltage-gated sodium channels of insects. And it causes paralysis and death unless they have mutations. And many, of course, evolve mutations, and many insects, of course, evolve resistance to insecticides. And that's why often you have to apply multiple insecticides or to use different insecticides each year. And a really cool scenario, so sometimes unexpected things happen once there's a toxin in an ecosystem. And in this case, we know, so this is a type of orchid bee, and this is a group of bees that often collect volatile chemicals, so chemicals that smell nice, that are present in the air. And so they'll often go to flowers and things and collect different sorts of chemicals and store them and use them as pheromones to attract mates. In this case, there's a species of orchid bee in Brazil where they still spray DDT on the side of buildings to prevent the proliferation of insects, of mosquitoes specifically. This orchid bee evolved for some reason to collect DDT and to use it as a pheromone. So I don't know that anyone has sequenced their voltage-gated sodium channel, but that's something I would love to do sometime, because they probably have resistance. So if you don't take anything else away from this talk, remember that I said toxins are awesome. There's so many cool things to learn about. From the perspective of humans, toxins have inspired many of our pharmaceutical drugs, including, for example, quinine, which is an anti-malarial drug that's produced by the cinchona bush that was originally discovered in Peru and had long been used by peoples in Peru. Penicillium, right? You might know of penicillin, but did you know that also lovastatin was described from this bacterium? And in fact, if you look over time, this is a graph showing the proportion of new drugs approved each year. This is the proportion of which were derived from natural products, so chemicals that were found in plants or animals. And it's a lot. It's around 40% every year. So, you know, nature is a great source of new pharmaceutical information. And I may have mentioned I love frogs. Frogs are also an underexplored source of interesting compounds. This is a psychological study asking people who had participated in a ceremony inhaling frog poisons, and they described a sustained enhancement of satisfaction with life. And I'll introduce you to this toad. So this is the Sonoran desert toad that was brought up by a member of the audience. So this is a toad that lives in the US and in Mexico. It's been used as a psychoactive drug because it produces 5-methoxy-DMT and also bufotenine, among other types of psychoactive compounds. For the frog itself, this is apparently sufficient to ward off predators. It's unknown, actually. I did quite a bit of reading to figure out if this has been, if this is a practice that's been done for a long time, but it's not really clear. What's clear is that other toads in the Americas have been used to kill people. So you can take toad toxins from the cane toad, for example, and distill it down, and it's a pretty effective poison. But what is known is that ceremonies with this toad are practiced currently by the Comcac Nation in Sonora, Mexico. And there's some really great videos online if you want to learn more. Another frog that has been used psychoactively is the waxy monkey tree frog. So this is a frog from the Amazon, and this is showing you generally how the practice is done. So they take the frog and they take the skin secretions. So I meant to say, in this species, so this is a toad, they have these huge glands right behind their eyes where they produce toxins. And in order to do this, basically what people do is it's called, I guess, milking, where you just kind of you put pressure on the glands, and the glands expel their contents. And then you would dry that, and you can reconstitute it or put it into a pipe and smoke it. For the waxy monkey tree frog, their psychoactive skin secretions are all over their body. It's not just from a single gland. And what has to be done is people will take a twig, a burning twig, and basically just open the skin up and then put it directly on the skin. And the reason why neither of these frogs are eaten is, in the case of toads, that psychoactive molecule would actually be digested by enzymes in your gut, so it's not psychoactive if you eat it. And in this case, it has to go directly into the bloodstream for it to be psychoactive. This waxy monkey tree frog, they have different types of proteins that are emetic, so they actually make you throw up. But they're also antimicrobial, and some of the ones that are used psychoactively are opioids. For the frog, again, apparently it's effective in preventing predation. This is something I'd be curious in learning more about. I don't quite understand how, producing a psychoactive effect in a predator is something that, I don't know. It's a very interesting question. In the Amazon, this frog has been used for a long time in ceremonies, mostly by hunters to increase stamina and resilience. Also to ward off bad luck. So the act of kind of throwing up everything is kind of a purging sort of feeling. And it's also historically been used as an abortifacient. and its name is Cambo locally. So, you know, I mentioned these frogs and the psychoactives in part because they're very interesting, and in many cases, these sorts of ceremonies have been practiced a long time by indigenous groups. And so if, you know, sometimes you can see these sorts of ceremonies offered here in the US or in other places, and if this is something that interests you, I just ask that you try to be respectful, that these are spiritual activities that have been practiced a long time by these groups. And the last talk in this series actually talks about this a little bit, and also how our community at UC Berkeley is beginning to work towards understanding the medical nature and spiritual practices of these compounds. So if you're interested, I encourage you to take a look at this talk. It's on the website. And lastly, I just want to mention, if these topics interest you or you think they would interest someone you know, my lab teaches a short course for high school students as part of Berkeley Splash. So this is an all-day event that takes place every semester. This semester, it's October 29th, and we'll teach a class called Animal Toxins in Natural History Museums, the Good, the Bad, and the Ugly. And so I'm a curator of herpetology, so of reptiles and amphibians in our Museum of Vertebrate Zoology. And we have vast collections of animals from all over the world. And so during this course, students can come and see the museum and to see the specimens and learn about toxicity. So if that's something that interests you, I encourage you to sign up. And with that, I'd just like to thank you for being here, and I can take in any additional questions. Yes, back there.
I know it's a little bit out of your field, but of course there's lots of toxic fungi. And I wonder if it's always the case in nature that somebody has figured out how to keep the toxic plant or animal or anything else. Every case you talked about, somebody has figured out how to get around it. I don't think that's always the case, or there's master toxins that basically nobody managed to deal with.
Okay, so the question was, many fungi have toxins, and of course lots of things have toxins. How is it that we've come to use some or not others? And so there's a very interesting paper that I read in preparing for this talk that talked about just that, which is that actually indigenous communities did a lot of the dangerous footwork for us. So there likely were many people who died trying to figure out what things can be eaten or not, and what things are psychoactive or not, and how to do them safely. So I think there's been a lot of kind of humans that have tried to figure this out, and from a Western scientific approach, there are certainly surveys of compounds where people will have a panel of activities they're looking for, so they might have a panel of molecular assays that show you, okay, this toxin would activate opioid receptors, or this one would block voltage-gated sodium channels. And so you might, for example, collect 30 types of sponges, extract different extracts, and then kind of apply them in different scenarios. So that would be like the Western modern approach. Yeah. I think did you have a question? Okay, so the question was, how did I start studying poison frogs? So it's really a confluence of events. So as a kid, I really liked frogs, and we had a creek in our backyard, and I used to play with bullfrogs and things. And then in college, I wanted to be a chemistry major because I had this really great chemist teacher in high school who really made me enthusiastic. But in college, I didn't do very well in chemistry, and then I was this is kind of a long-winded answer, but I was outside a biology lab, and I saw an article on the wall that was like, you know, Professor so-and-so just got back from the Caribbean studying lizards. And I realized, like, actually, biology offers a lot of interesting careers that are focused on the animals. And in graduate school, I knew I wanted to work on frogs, And by that time, I had lived in Ecuador for some time, so I knew I wanted to work with frogs in Ecuador. And then I also knew that I wanted to work in different fields of biology. So I wanted to do, not just mechanism and not just ecology, but a project that did those. And for me, working with toxins in frogs kind of hit all those marks. Yeah. Yeah. Yes. Okay, the question is Phyllobates terribilis, the golden poison frog, the same as the golden toad from Costa Rica that's extinct? No, it's a different species. They're both toxic. So the golden poison frog has toxin, and the golden toad has tetrodotoxin. So the question is, there's a group of salamanders here called the slender salamanders, and the genus, the scientific name is Batrachoceps. And the question was if that has anything to do with batrachotoxin. No, it doesn't, but they share the same root word, which is, I think, batra. I should know what that means, but I don't know off the top of my head. Yeah.
Just read Darwin's Voyage of People. I really enjoyed it. When I'm reading Origin of Species, I'm finding how boring, but the trip around the world. I don't remember reading a conference author there. Did he publish it, or how do we know about it?
Okay, so the question is, how do we know about this beetle story of Darwin? I think it's in one of his letters. Yeah, but I actually helped write a TEDx video that describes this, and I think it's on the list of sources. So if you're super interested, you can look at the video on my website, and there might be a list. Or you can email me, and I'm happy to send it to you. Other questions? Yes. Yeah.
Oh, so I have like 2 questions. So like, I remember you saying like those poison dart frogs, they get like toxins from like the supply of beetles. Like, where does beetles get those toxins from? What sources? And also like, I remember you saying like this poison frog like develop convergent evolutions. And if all of those like 3 different like branches have like same sources of like toxins, I'm kind of curious.
Yes, okay, so the first question was, we know that poison frogs get their toxins from what they eat, and in some cases we know what insects or mites are the sources, but where do those insects or mites get it? And that we don't have an answer to. So there's a couple studies that show definitively that ants, beetles, and mites have the same exact compounds that we find in the frogs, and that the frogs eat those ants, mites, and beetles. So we know that that's at least the first step. We don't know, I think in any of those cases, if the mite, the beetle, or the ant is producing the toxin. I know that mites are important sources of this type of toxin, so mites could be an ultimate source. In the case of the beetles and the ants, it's not clear. It could be a bacteria living on the ant. It could be a bacteria living in the ant colony. It could be a fungus that the ant cultivates, because some of the ones are these leaf-cutting ants. It could be a bacteria living on the fungus that's cultivated. We don't really know. It's an open question. And then your second question, if I remember, was that toxicity has evolved several times in poison frogs. Do they get it from the same thing? And the answer is yes. What's interesting is that all of these frogs, poisonous or not, are eating the same sorts of food, and really what's different is their physiology and kind of how they metabolize the toxins or not. And what's even cooler is there's a different type of frog in Madagascar that looks really similar, eats similar things, and has the same toxins. And so what's awesome is that, insects are this very old, ancient group of animals where you might see closely related insects on different continents. producing or obtaining or having a symbiosis to have those same compounds. And so you get then frogs in South America and frogs in Africa sequestering the same compounds from those different species. And one other quick anecdote, someone in Hawaii thought it would be a good idea to introduce poison frogs onto their hotel lawn. They're really pretty, so I understand. And so if you go and you collect those frogs, which are not native to Hawaii, they also have the same sorts of compounds in their skin. So it's really cool. Any other questions? Yes?
So I want to ask about the fire ants and like the cytotoxins that they produce.
Yes.
So, how do fire ants specifically use cytotoxins? Is it just like for a defense mechanism that's like made more like, powerful by the fact that they're in like a big group or something else?
Yes. Okay, the question was, how do fire ants use their venom and why? And so some of it certainly is a defensive mechanism. So if they feel afraid, they can group up and attack something. But they also can use it for hunting. So I just saw a talk recently about, it was lizards. So they had this big area where they could fence off and they could like capture all the lizards. And it happened to be an area where there were fire ants. And in one plot, they killed all the fire ant colonies. In another plot, they left them. And what the fire ants will do is they'll gang up on a single lizard, and like hundreds of them will sting the lizard, enough so that the venom load is so high the lizard would die. And then the fire ants will eat the lizard. Yeah. I think we're about out of time. Is there one more? I can take one more question if there's one. Okay.
Such an active crowd. Give Becca one more round of applause. Thank you everyone for being here. I just want to thank Becca one more time with the Science at Cal shirt. So as a gesture of our thanks.
Thank you.
And have a good weekend, everyone. If you want to talk to her, she'll be up here to learn more. I also just wanted to say that there were so many, like, we don't know answers, which means that all the young scientists in the room, you have a lot of work to do. Not to put any pressure on you, but